Morning. Today is Monday, June 1st, 2020. My name is Ricardo Sandoval, and this video is for week 3 of Biology 225, Microbiology at Los Angeles Pacific University. This is my results video for lab number 5, which is entitled Bacterial Morphology and Staining Techniques. And then here's just a quick little sign I made with the information I just spoke of. So in this week's video, what I did was I learned about gram staining, gram-positive and gram-negative organisms and how to tell the difference. So I made slides of my cheek cells, my tartar or plaque, as well. I used previous organisms I had cultured of E. coli, S. epidermis, and S. cerevisiae to go through the 4-step process that entails gram staining. So throughout the entire exercise or laboratory, however you'd like to call it, I didn't really experience any problems. It was very interesting to see how the peptidoglycan in gram-positive organisms does hold the crystal violet in place. So because I used crystal violet by itself, Congo red by itself, and then of course I used for the E. coli, S. cerevisiae, and S. epidermis, the 4-step process of crystal violet, PDP iodine, the decolorizer, and then the last portion, the safranin red, just to determine whether or not the organisms themselves were gram-positive or gram-negative. So when it came to just doing the direct staining technique that I just did for the cheek and for the dental plaque tartar, it was just a drop of the crystal violet and then a drop of the Congo red, but on separate slides, and it was very easy to visualize. Excuse me. The slides themselves, or the organisms themselves, under the microscope. Now, when it came to doing the 4-step process on the E. coli, the S. epidermis, and the S. cerevisiae, what I learned, of course, is cells that, or bacteria rather, that are gram-positive stain purple or violet because the peptidoglycan, or those organisms, have a very thick wall of peptidoglycan that holds the crystal violet in place, while gram-negative organisms don't hold the crystal violet in place and tend to stain pink, which is the color that is developed by the safranin red, which is the last stain that I put in. So as far as organisms are concerned, of course, there's gram-positive and gram-negative, and in medicine, there's a lot of talk about gram-positive and gram-negative, and how those two organisms are treated is a bit different. So what was a bit surprising to me is that gram-positive organisms or gram-positive bacteria are much more easier to treat using antibiotics versus a gram-negative organism because of that thick wall of the peptidoglycan. So if you were to, let's say, have cellulitis, and it turns out that this particular version of cellulitis, which is a skin infection, is gram-positive, and you're prescribed clindamycin, let's say, or Keflex, 2 common antibiotics. The thick wall of the peptidoglycan will pretty much hold onto or strongly absorb the antibiotic, which leads to death of bacteria, death of the cell, and then eventually phagocytosis. Versus a gram-negative organism that has a much thinner wall of peptidoglycan, and therefore it doesn't hold the antibiotic in place as easily. So curing that bacteria takes a bit longer. This is also important when it comes to something called culture and sensitivity. So for example, for a patient who has sepsis, who is septic, who has a whole body infection, that patient starts out with a very broad spectrum antibiotic like Rocephin or vancomycin, let's say. just to kind of start doing something. But because those two antibiotics are very broad spectrum, very wide, they can cause damage to the body versus just attacking that one organism or that one bacteria that is harming the body. So in culture and sensitivity, of course, a culture or a sample of the bacteria is grown, and then certain antibiotics are exposed to the bacteria to see which one works best. But again, in that respect, a gram-positive bacteria has a much more likely or faster turnaround, if you will, of being cured versus a gram-negative bacteria. So if a person who is septic, who is already very, very ill, has a gram-positive bacteria and they stay on vancomycin or go on clindamycin or Keflex or one of the many antibiotics out there, for example, Bactrim, that medication has a much stronger likelihood of curing them first time around rather than a gram-negative bacteria that because of the lack of that thick peptidoglycan cell may not work as efficiently and may require other medications to help with that process. So again, just to talk about the four-step process of gram staining. So we start with crystal violet, rinse that with water, then go to the PVP iodine, rinse that with water. The decolorizer dehydrates the stain and removes any errant dyes that aren't being held on by the bacteria itself. And then lastly, of course, is the safranin red. So again, gram-positive will stain the violet color. Gram-negative will stain pink. Now, if there's any other color variation in there, there may be a combination of gram-positive, gram-negative, or other organisms, or there may have been a problem with the collection of that culture or that specimen. So with that being said, this will be the conclusion of my video. Thank you.